much. We will now invite Ricardo Arminsen from Chile. He will be talking about the challenges of implementing genomic medicine in Latin America. So Ricardo, all yours.
Thank you very much. Next slide, please. So, well, thank you everybody for being part of this meeting, and also for the opportunity to open the discussions about the local and regional challenges that we face about implementing genomic medicine. Also, I hope this is not the end of the conversation, it's the beginning. We will hope to have a face-to-face meeting next year. So we're just gonna lay out a few topics for future conversation. Next, please. So I recommend everybody to go over this very nice book from 2018 by Catalina and George and many other authors, where I think they did a very nice job identifying and systematizing all the challenges that we can face, especially in developing countries. And as you can see in the balloons, the word lack, it's very often. We have lack of many abilities, many skills, data, policy, regulation, of course, funding, and many others. So I wanted to take a step back from these very specific challenges and see what's the ground where this genomic medicine implementation should occur in Latin America. Next slide, please. So I think maybe you already saw this map. This is the global domestic product per capita and each of the countries over the world. And you can see Latin America is in the middle to low end. Maybe Chile has a slightly higher global domestic product than the other countries. But as I mentioned, we are in the middle to low end. And next slide, you can see this is not only about the average income that people will have. Also, if you look at the economic inequality that the Gini index revealed, you can see many of the South American countries are not doing so well. Especially the high income ones are not doing very well in this Gini index. So average are hiding the distribution of the income in these countries. Next, please. And at the end, if we see how much effort are we doing in research and development, well, you can see again that we're not doing much, actually. The opportunity to grow, how much we spend on research and development is there. And this brings my first thought about this. And I think we need to, if we want to move forward with the implementation of genomic medicine in Latin America, we need to really challenge ourselves and make an extra effort to highlight what is the cost effectiveness of genomic medicine for a specific disease, conditions, for prevention, and for treatment. Because in this low resource setting, every dollar, every peso really counts and has to compete with many other needs. So we really need to make a very good case about the use of genomic medicine and not take it for granted. And this is going to mean extra effort for all of us. Next, please. Also, I think we're in a complicated setting. I think you've seen this missing diversity paper many times. This highlights the fact that Hispanic or Latin American people is very not represented in studies and databases, at least at the genome-wide level. Next, please. And just to remind you that Latin America actually is a very nice mixed population from Europe, Latin American and other minus components from Africa and Asia. And also is very different across different countries. You can see in your left the data from 23andMe and across different countries, but also in the map on your right, you can see that native populations are very complex. We have many of them. Some of them have been very isolated for many years, so they also add complexity to the admixtures and to the genetics of our population, and probably has an impact on how we make good use of genomic medicine across this different population. Next, please. So if you look at this is the Genome-Wide Association Diversity Monitor. You can see that over time, we're not really improving much the participation of Hispanic or Latin America is not very high, and it's not really getting better. Next, please. Okay. Now, if we look at the 2015 status of the 1,000 Genomes Initiative, you can see that also Latin America was not very well represented, with the exception from folks from Colombia and Peru. Probably this has improved over the last 5 years, but as you can see, at that time we were not really contributing much to the genomic information about the diversity across the world. So this is something that probably, as we learn from the Brazilian Genome Initiative, is improving. And in the next slide, you can see there are many local and regional initiatives on human genomic data from Argentina, Uruguay, Ecuador, Brazil, also in Chile, and many others. I apologize if I am not mentioning your initiative, but I think this may improve over time. But what strikes me is that many of these initiatives are happening at the local arena for each country, and I think we really need to think in a broader perspective and consider the possibility of collaboration across countries. Something like Latin Gen, I believe, is a very good initiative, not only focusing in one country. Next, please. And then, of course, there is insufficient genomic knowledge. This is not only true for Latin America, probably it's true for many other regions of the world. And we have many efforts, like this Latin American School of Human and Medical Genetics that goes every 1 or 2 years and is open for many participants from Latin America. Also in Chile, we make a special effort to develop biobanks since 2013. We're hosting international symposiums in biobanking for research and also for implementation of genomic medicine. Next. Okay, and also we got support from the National Cancer Institute from the United States to do I think 3 versions of an international workshop in cancer genomics and bioinformatics. Also, we try to highlight the genomic diversity and how this affects personalized medicine and cancer research and implementation of precision medicine in oncology. So we have some local efforts that I think are worth describing, but of course there is still a lot more to do, and I believe international collaboration, and now with Zoom and all these virtual meetings, that's really an opportunity for further training and outreach to our Latin American community. Next, please. Okay, so with the I just wanna when I get to this point in my talk, I say, well, I say to myself, where do we go from here? And then I learned there are movies, there are books that are addressing this same question. And I like to bring examples of many of them, because I really think with the pandemic, we had the opportunity to rethink the way we're doing the implementation of genomic medicine in Latin America. And maybe we not only need to think outside the box, maybe we also need to think, take the box upside down, destroy the box, or maybe burn the box, and really bring new ideas to the table from many areas, from a movie from the 50s, from Martin Luther King book, from Bernie Sanders, and from many others, to really take advantage of the situation we're facing now and do things in a different way. Next, please. And I think if we look at the way biotechnology, genomics, and medicine is organized around the world, and we take as example different cities like California, like Massachusetts, like Washington, like San Francisco, and also places in Asia and Europe, you see a number of academia, institutions, the government, biotech companies, health providers working together in a cycle of research, development, innovation, and implementation. And so far, with the exception probably of Brazil and Mexico, Latin America is just playing the role of, or most of the time is playing the role of a customer. And I think we need to step away from that customer role. Of course, it's great to take advantage of the technologies that are available from other parts of the world, but also we need to become active players in this ecosystem of innovation, research, and development, because this is not going to happen only through increasing the funding, but also we need to be part of the value cycle And I hope that's going to be a way to get implementation of genomic medicine close to our patients. Next, please. So I want to acknowledge the Chilean government. We got support from many of the grant agencies in Chile, I believe probably from all of them, also from the National Cancer Institute, also from Corfo, that's the economy office in Chile. Also, we had support from Pfizer and Thermo Fisher, two transnational companies that invest in South America to develop research and development locally. Thank you very much.
Well, thank you so much, Ricardo. I think there are many things we can discuss at the Q&A session, but two key messages for me, and even more now that I am in Colombia. And I think one is we need to work closely together. We see that Europe, the US, Canada, they collaborate, they work together. And here in the South, in Africa or Latin America, we work, each of the country works independently. So I think collaboration in these regions is important. And the other one, going from being a customer to being, even a producer, I think is a very important message. So thanks for that. And now my pleasure to introduce a person that doesn't need any introduction. Of course, Jeff Ginsburg will be talking about the Ignite initiative. So all yours, the floor, Jeff.
Thank you, Catalina. Thanks, everybody. So yes, I'm going to talk about Ignite, which is an acronym for Implementing Genomics in Practice. It's a national network in the United States. Can I have the next slide, please? I wanted to start off with a quote from Thomas Edison, who is arguably one of the world's greatest inventors, and he said, "Vision without implementation is hallucination." So Edison was a very meticulous planner, and I think it should be the same case with genomic medicine. We cannot leave implementation to chance. We need to plan implementation carefully, otherwise it may fail. So next slide, please. The Framingham Risk Score, which may be something familiar to hopefully everybody on this in this conference, I would argue was initially a failure to implement. In 1961, the first factors of risk for heart disease were published, and you can see them here. Next slide, please. But 50 years later, it was really seldomly used in primary care. You can see that primary care physicians, actually a very small percentage of them, actually read guidelines carefully. A slightly greater percentage of them actually used a coronary heart disease risk factor calculator in their practice, and they attributed this to variability in the knowledge, beliefs, and practice patterns. The authors of this paper attributed this to the variability in these things, and physicians, rightly said that there was a lack of knowledge, of distrust in the validity of these things, and it was time-consuming. I would say that the electronic medical record has changed a lot of that for this particular tool, because EMRs now can deliver the Framingham Risk Score without the need for a lot of intervention by the practitioners. But still, many other tools may fail if there's not a proper implementation strategy. Next slide, please. The other thing that's been a challenge for implementation of genomic medicine has been the lack of appropriate funding for what is called T3 research, or implementation science research. These are data from about 10 years ago from the National Cancer Institute, as well as the National Heart, Lung, and Blood Institute from the National Institutes of Health in the United States, which showed that the vast majority of funded applications in genomics research were really focused on the earlier stages of discovery science, and very few, less than 2%, were focused on implementation and outcomes research. Next slide, please. The other issue, again, in the United States is that like it says here, you've seen one payer, you've seen one payer. Not all payers are alike. The payer adoption of the genome-based technologies and reimbursement is very slow. On the left-hand side is the adoption curve for the Oncotype DX test for breast cancer prognosis. And you can see it took over a period of over 6 years for large-scale payer adoption and reimbursement of that test. On the right-hand side of this slide are coverage policies for a number of different United States payers across a variety of cancer-related tests. And you can see, based on the check marks in the table, not only is there payer heterogeneity for a particular test, but even discordance between FDA clearance and payer adoption. And this leads to both confusion as well as a stagnation of implementation. The paper at the bottom highlights work that we did just a couple of years ago, which essentially recapitulated the fact that these technologies are still reimbursed on a heterogeneous basis. Next slide, please. So it was on this background that in 2013, the IGNITE network was formed in the United States, sponsored by the National Human Genome Research Institute at the NIH. Its goals were to expand and link existing genome-based efforts in genomic medicine, to develop the implementation methods in diverse settings and populations, to contribute to the evidence base regarding outcomes of incorporation of genomic information into clinical care, and then to disseminate best practices for genomic medicine implementation, diffusion, and sustainability. Next slide, please. The network was composed of 6 sites and a coordinating center. Our site at Duke was the coordinating center, and each site had multiple clinics, so there were approximately 30 or more clinical care settings across the United States participating in the first phase of IGNITE. Next slide, please. I just wanted to highlight just a couple of examples. Time will not let me provide you many more of some of the results of implementation of this program. In this particular, in one of the projects in IGNITE was a family history implementation project in diverse care settings. And the results indicated here with a proper family history show a significant percentage of the general primary care population meet criteria for intensive risk management, with approximately 30% of people being identified at high risk for monogenic disorders. 27% received recommendations for screening and prevention of familial cancers, and 56% received recommendations for chronic disease dominated, in this case, by diabetes. In the absence of a properly implemented family history, very few of these individuals who actually would have been identified as being at risk. Next slide, please. Another project in the early IGNITE network was the implementation of pharmacogenetic testing for cytochrome P2C19 in the setting of cardiac interventions, percutaneous interventions, which showed that individuals who were on clopidogrel, who carried a loss of function mutation, had higher incidences of major adverse cardiac events, or MACE, than those without the loss of function mutation or those who were on alternative medicines. Both of these observations, the family history and the pharmacogenetics implementation, illustrate what can happen when genomic tools are actually properly implemented. Next slide, please. So what did we learn in the first 5 years of IGNITE? We learned that implementation is a science, and it's actually hard. There are complex clinical workflows that must be understood, and they actually are very different in diverse settings. That we need to actually have, in addition to clinical and other types of measures, we need to have process measures that actually allow us to optimize the integration of genome-based tools into patient and provider workflows. Electronic medical records are not ready to incorporate genomic data. Local clinical champions are key, and having a network is a very powerful way to carry out these kinds of studies. And finally, that genomic medicine research, which actually engages underserved minorities, can actually help reduce disparities as part of the research agenda. Next slide, please. In 2018, we began IGNITE 2. This is a genomics pragmatic clinical trials network. Some of the elements of this slide are missing, but I'll fill in the blanks for you. This is a genomic pragmatic clinical trials network focused on measuring effectiveness of genomic interventions. The paradigm is to randomize patients in a primary care or specialty care setting to either receive standard of care and measure their outcomes, or to receive genomics guided care and also measure outcomes. The focus of the IGNITE 2 network is on pragmatic clinical trials in pharmacogenetics, as well as chronic disease risk in diverse population. And we are measuring the outcomes both at the patient, provider, system, as well as economics. Next slide, please. We have 3 planned pharmacogenetic trials under the umbrella of what's called the ADOPT PGX. So you can see what the acronym here stands for: Depression and Opioid Pragmatic Trial in Pharmacogenetics. The goal is to determine whether genotype-guided therapy of opioids and SSRIs leads to improved acute and chronic pain control, as well as control of depressive symptoms. The diagram on the right, which I'm not going to go through, illustrates some of the complexity of the trial design of each of the 3 trial designs, but the basic premise for all of these is that the intervention population receives genetic information up front, the providers receive genetic information up front, and the control population receive their genetic information at the end of the trial. Next slide, please. The other trial in the IGNITE-2 network is called GUARD, which stands for Genetic Testing to Understand Renal Disease and Disparities Across the U.S. And this is to determine the effect of participant and provider knowledge of having a positive ApoL1 allele on blood pressure management and change in systolic blood pressure after 3 months. Again, individuals were randomized to receive genetic testing up front or to receive it at the end of the trial. And the outcome measures are, as I said, measurement of a change in systolic blood pressure at the end of 3 months. Next slide, please. So we were to have started enrollment in these pragmatic trials in March, but then COVID-19 appeared and we had to put all on hold, which sort of gives us all this other lesson learned from pragmatic clinical trials, is that things do change in the real world and impact the original study design. And we're actually in the process of re-engineering some elements of these trials to be done through teleresearch or telemedicine. Next slide, please. So finally, I just wanted to close with some thoughts about lessons perhaps from IGNITE, and that we can discuss in the panel discussion on what the opportunities are to implement in low and middle income countries. I think this has been mentioned throughout this conference and in others, but there certainly is a need to build the proper genomics capacity, both in terms of the laboratories and the workflows in these regions, as well as the competencies in genomic knowledge, which I know Rich Haspel will be talking about in the next talk, along lines also to emphasize our ability to communicate and make decisions across a vibrant genomics workforce. It's clear that we need to have better population literacy and numeracy in order so that we can better engage our research participants. And as I said earlier, the diversity of practice environments is going to require a diversity of implementation strategies. And as has been said many times in the last 2 days, there's an enormous need for collaboration among multidisciplinary teams. So I would sum it up in saying the G2MC can ignite the LMICs. Last slide, please. And finally, I just wanted to thank the constituent members of the IGNITE network. I think some of the logos here have not reproduced well, but it's an enormous amount of teamwork and a great team. And again, thank you for allowing me to share my thoughts about IGNITE with the G2MC.
Yeah, thank you, Jeff. And clearly, you know, this great work shows what you say during the presentation, which is that implementing genomics in medicine is not a trivial thing. It needs a whole process and methods, and you clearly are showing this with the great job IGNITE is doing. So thanks for the presentation. We'll go into questions at the end of the panel. Now we have the presentation from Rich Haspel. He will be talking about the ISCC, which is a collaborative approach to education. So all yours, Rich.
Thanks. I really appreciate the opportunity to talk a little bit about ISCC PEG, because this is about solutions to roadblocks, and ISCCC is all about education, which is usually a part of a solution to a lot of different roadblocks. So first is, what is ISCC PEG? Well, it's a very long acronym for Inter-Society Coordinating Committee for Practitioner Education in Genomics. It was formed in 2013 from the Genomic Medicine 4 meeting, and it's supported by the NHGRI, Division of Policy Communications and Education, and specifically the Education and Community Involvement Branch. And two very important people who help with ISCC from NHGRI are Carla Easter, who's the chief of ECIB and is also the co-chair of ISCC, along with myself, and Donna Messersmith, who is a provider education specialist at NHGRI and plays a very important role in organizing ISCC. So ISCC is a committee, and it's made up of currently over 150 members. The initial format of ISCC was to have representatives from organizations and NIH institutes. But since that time, we now have representatives from over 80 organizations. But more recently, we've invited both individual members and also industry members. So, and we're always looking for new members. And we are international, but you know, you don't have to be in the US to join this, even though it's NHGRI. But we could definitely use more international presence in our group. So this is a slide which was, I have to give credit to my daughter Lily, who's 14, who helped put it together. But it's basically showing all the different organizations and institutes and groups that are part of ISCC. And you can notice there's a bunch of NIH institutes. There are a bunch of different societies and organizations. There are several companies and think tank type groups. So the idea is we really have tried to build a collaborative group with a lot of different perspectives. And again, not just genetics organizations. These are organizations like the American Academy of Pediatrics, the American Heart Association. So just all types of, and not just physicians. We have physician assistants and also genetic counselors and nurses and all sorts of healthcare providers who are interested in education in genetics and genomics. So how does it work? Well, there are two co-chairs. One is an NHGRI co-chair, which is Carla Easter, and the other one is me. And as I mentioned, there are different types of members. There are representing an organization or an institute, individuals, and industry. And we have some policies, such as we want to ensure independence. That is like trying to avoid conflict of interest and other things. There are no dues. Everything is completely free. We have conference calls every other month where members provide intros and updates, and we talk about some initiatives. For example, one initiative now is we're trying to create an ISCC Scholars Program where we have a student who we support going to meetings and things like that and working on a project. So we're discussing how to get that going. And we have project groups, which I'll talk a little bit more about. We also have an in-person meeting once a year. It's held at NIH. We ask that individuals fund themselves or through their organization, but people can call in if they can't come in person. And we have a website. It's in the lower right over there where we provide a lot of information. And also the project groups have conference calls. And I mentioned I'll talk a little bit about that. And we also have a compendium, which is a list of all the different genetics, genomics related activities that our different organizations or individuals are doing. And I'll show that in a sec too. All right, so the goal of ISCC is to improve genomic literacy, in particular among healthcare providers. So under the general goal of collaboration, we want to determine the needs for education and create useful resources. And we also want to disseminate and learn about best practices. One thing we tried to do with ISCC, this is one of my favorite demotivation posters, committees just like teamwork, only without the work. We try to actually not just sit about and talk about things, we also try to get things done to help people learn about genomics. So here are some resources. We've created competencies for genomics. We've created different actual teaching tools, including webinars, cases, and also contributing to some existing projects. So for example, we created these things called universal genomics exercises, which are basically plug and play team-based learning exercises that any specialty can use, put in their genes, put in their diseases, and it helps the learner use online genomics tools related to those diseases. And we've had workshops using these at the American Heart Association, the American Academy of Ophthalmology, and the American Academy of Neurology. We have a project group that's worked on something called Project Inclusive, which is a implicit bias module related to prenatal screening and disability, which Shamita Dasgupta, who's running that group, is doing a study using that module. And we've contributed to something called G3C, which is a series of online modules for teaching. So I mentioned the compendium. This is just basically, you can get this online at our website. It lists all the resources for organizations or individuals or industry who wanted to list them. We have for each of them, it lists things like, their active educational goals, what they might be interested in working with ISCC and things like that. So if you have a chance, you can take a look. And then I mentioned, where do these resources we create come from? We have project groups, which aren't just talking about the problems in education, but actually trying to figure out ways to solve them. We have the inclusion and practice of genomic medicine I mentioned, which is created a module on, as I mentioned, on implicit bias and disability. We have a pharmacogenomics work group, which is working with the American Academy of Family Physicians to create online resources and cases. We have a direct-to-consumer genetic testing group, which is working on a sort of an FAQ to help primary care providers navigate through direct-to-consumer testing. And we have a rare diseases group, which is looking into ways to kind of thinking of leveraging those universal exercises to teach about rare diseases. And we clearly have interest in G2MC about rare diseases based on the session earlier this morning and some of these other groups. So the key thing is, we welcome new members to ISCC or project groups or new ideas for project groups. We just had one presented to create an obstetrics and gynecology genomics curriculum for residents and fellows in OBGYN. And there might be opportunities for collaboration, because I'm also co-chair of the education working group for G2MC. So are there ways we can collaborate across the groups? So just to summarize, ISCC PEG is a way that we're trying to use education as a solution to help improve genomic literacy and care of patients. We have a diverse and large membership, organization, institutes, or if you just are someone interested in genomics education, you're welcome to join. Again, it is focused, however, on healthcare providers. It's not only a form of discussion, but a way to develop collaborative resources. We have new members and project groups are always welcome, including international. We'd really love to build up the international presence with our group. And we're continuing to think of ways for the two groups to collaborate. And just a lot of thanks, of course, to NHGRI, who supports ISCC PEG, Carla Easter and Donna, who are critical for keeping ISCC going, and also Christina Dalton and Charisse, who are part of NHGRI and help with ISCC PEG activities. Laura Rodriguez was head of the division that oversees ISCC PEG, and now it's Larry Brody who does that, so thanks to him. And Terry Manolio, who you all know, has been very involved in ISCC PEG. She was co-chair with me and still provides very, very helpful advice, so thanks to her as well. Everyone who's a member of ISCC PEG, and of course G2MC, which is a member of ISCC PEG, and for giving me the opportunity to talk a little bit about it. So that's what I have, and again, thanks for the opportunity.
Well, thank you, Rich. That really illustrates the efforts that are being made on education. I think it's so important. We have been talking all through these 2 days about how important it is, and even more important, I would say, for developing countries. And I know many of us in the South are trying to implement some of these initiatives. So thanks for that. We will go into questions at the end of the panel. And now we go with Phil Febo from Illumina, talking about the next generation sequencing in limited resource settings. So all yours, Phil.
Thank you, Catalina. And I really appreciate the opportunity to join today. So, it's an honor to be able to participate in this G2MC conference today. It would have been great to join you all in Chile. I really appreciate the organizers' creativity in leveraging Zoom and on resources to make sure that we're still getting together and talking about it, albeit virtually. I get to talk about Illumina. And, you know, I was drawn to Illumina because the mission of the company is to unlock the power of the genome to improve human health. And, you know, we are a global company, and increasingly we understand that unlocking the human genome has to be available across the globe and in countries with limited resources, because not only does that make us all more informed, does not make us all have a better information about the human genome and all its diversity. It's really the only way we're going to unlock the full power of the genome, by getting full representation and involvement. And also, it's also a imperative to bring that to populations that have limited resources, and to individual patients who are working within health systems that are strained and constrained. So I don't have to talk to this group about the different ways in which genomics is having an increasing impact on human health. But in the time of COVID, I thought I'd walk through some of what we're doing through the lens of infectious disease, and then end, apropos to earlier discussions today, on some examples from rare disease and genetic disease. So, you know, as we find ourselves, you know, in isolation because of COVID, it's really important that we understand, you know, sequencing is nothing other than a way to read out genomes. And in the case of infectious disease, those genomes can be DNA-based or RNA-based. COVID is an RNA-based virus. And that's what our technology reads and provides insight to. And, you know, at the end of the day, Illumina is a technology provider, and it's the folks on this call and the folks who work with G2MC that are charged with figuring out how to best bring technologies into different environments to improve outcomes. But what we have the obligation to do at Illumina is make sure that we have technologies that can serve that purpose, that serve in the purpose of health, and that we have a range of technologies to accommodate a range of capacities. And so, you know, as we've worked to respond to the challenge of COVID, and as I've engaged with investigators across a broad range of settings, both in countries with significant resources and those with countries with limited resources, you come to understand that one size does not fit all. And so certainly you have to start with patient sample collection. You have to figure out how to extract RNA. You know, already we're seeing solutions that are very inexpensive, where you can collect a sample, you can isolate RNA. But as far as getting to sequencing and understanding what virus is of it, what is present, confirm that perhaps it's COVID or perhaps an influenza virus, that you have to have different options for sequencing. So there's different methods. There's shotgun methods where you sequence everything, including host and whatever other genomes are included. There's enrichment, where you're specifically pulling out specific viruses. There's also application-based sequencing, and certainly there's a range. Illumina is not the only sequencing company, and there's some others that have, you know, simple point, you know, sequencers that may be easier to bring to point of care. But in our fleet, we have a range of capability from a very high end and high capacity to more modest size, something that's the size of a large toaster, the iSeq. And after getting the sequencing performed, you need the informatics. And increasingly, we're working towards making those informatics more approachable, because as you think about this walk, as you think about the capabilities required in country, in a lab to do this, many of the upfront work to prepare the sample is broadly available. Our sequencers are increasingly available, and some of you may have seen, we've been working very hard to donate sequencers to regions that are asking for help in the setting of COVID. But then you get to the information and the bioinformatics, and I've heard a lot of concern about access to bioinformatic expertise to analyze the data. So we do see it as part of our mandate to make that more approachable and have the bioinformatics and a toolkit that is accessible and doesn't require deep bioinformatic expertise in order to identify a virus, identify the sequences, submit to sharing collaborations such as GISAID, so that we can track this virus globally and track infection. So we are committed to figuring out how to make sure that there is an opportunity across the spectrum of capabilities to leverage the benefit of sequencing. This is a complicated slide. I'm not going to spend the time to walk through, but it certainly talks through the different capabilities, the different time, so that just to make sure that depending on what the lab's capabilities, what the region's capabilities are, we try to find a way to make sure there's the right capability. And I do want to talk about a story that predated COVID to emphasize this point. And this has to do with the Ebola outbreak in the Democratic Republic of Congo. And this outbreak, there have been multiple outbreaks, but we've been working with USAMRID to enable them to go into region and to really understand these outbreaks. And specifically for Ebola, with multiple introductions from zoonotic host, it's really important to understand, is an outbreak new? Is it an outbreak contained? And again, for the sake of time, I won't walk through the full timeline. But with the iSeq now, investigators were able to check a sequencer on their flight to the Democratic Republic of Congo. They worked over a period of about 4 to 5 weeks to train laboratory scientists in the country with a pretty rudimentary lab on how to use the iSeq, and and they were shortly able to independently run the sequencer. They ended up using the sequencer for Ebola. They also used the sequencer for monkeypox, which is also endemic to the region. And interestingly, despite there being containment of one Ebola outbreak by July of 2018, immediately there was another case in a region close by, and they had to determine, is it the same outbreak or was it a new outbreak? Was there a lack of containment or was it a new outbreak? And they were able to do the sequencing, the DNC laboratory folks, and they confirmed that in fact it was a new outbreak. It wasn't a lack of containment. It's just an example of how with the right technology, understanding some of the limitations, you can empower broadly. And as we think of other infectious disease examples where access to sequencing is becoming increasingly important, certainly tuberculosis is a very good example where with relatively limited sequencing now, laboratories can determine not only the presence of the tuberculosis, but also determine the drug sensitivities. Sequencing is now adopted, for example, at the New York State Health Department as the method of choice to determine the drug sensitivity. And I think increasingly we'll see entities, when they're empowered, move away from culture for sensitivity towards sequencing-based methods because the tuberculosis genome is relatively small, and you really can map sensitivities based on the variants identified. Obviously, surveillance is front of mind for a lot of entities. The global pandemic that we're in is a bit of a demonstration that while we do have great capacity and we have great technologies, we're not set up and prepared yet to respond rapidly enough to potential pandemics. And so we need to do more work. And I do think, as we heard earlier, the combination of neglect that happens in between pandemics, we have to move beyond. And we're dedicated, and part of what we're doing in providing our sequencers in the time of COVID is shoring up capabilities so that we can do a better job of proactive surveillance and enable that. Again, for the sake of time, I don't want to walk through it, but we've been very proud with the role that sequencing's played in COVID, sequencing, identifying the virus, showing that it probably came from a bat host, and all the efforts from hundreds of labs across the globe that have been contributing to GISAID to let us track this, followed over time, understand it, and we're also seeing the evolution of the virus because of sequencing. And this, 10 years ago with H1N1, this was not possible. It was too expensive. It wasn't accessible enough. I think some of the benefit we're seeing now has to do with the fact that it's become more affordable, more accessible. But we have a lot of work to go, and specifically with the goal of empowering countries with less resources. We do have more work to go. This maps the cost of a genome going down from the initial Human Genome Project down to now $1,000. In many places can get less than $1,000. This is a human genome, and we're seeing many hundreds of thousands of genomes sequenced. Again, apropos earlier comments, many of these genomes are from the developed world. They don't adequately represent the diversity of our human race, and we really have to do much better in enabling areas with ethnic diversity, with populations that aren't represented, in order to get to a more complete understanding of the genome. And this is a complicated slide, but one of the elements is that bioinformatics concern and computational capabilities. The model often in the past has been to have your servers for your sequencers on site. Increasingly, this is shifting to the cloud. And I think that is empowering. Obviously, not all areas, not all countries have a robust cloud infrastructure, but that will change over time. We will see that build, and it will decrease the challenge to invest in on-site infrastructure. And I think that will be quite empowering in these countries. And just in my closing comments, I do want to talk about rare and undiagnosed genetic disease, because a whole genome has now been shown, certainly in developed countries, to provide the answer to these children and these families. When I put together these slides, I was a little concerned, well, does it make sense to talk about whole genomes in countries of less resource? But it absolutely does. And we have a very good example at Illumina. We've had the iHope network, which brings together labs and even, and make sure families that couldn't afford a whole genome for their child, because there's still a lack of coverage even in developed countries, have access to that without charge. And we've actually worked very closely with a hospital in Tijuana, California, Mexico. And Dr. Marilyn Jones and Diane Macer-Fry have been running a clinic where families bring children about once a week for evaluation. The data from that is incredible in that we've enabled them to order whole genomes. We've performed 60 whole genomes on children who are brought in. About 68% of the time, the diagnosis is determined with the whole genome. And really important to today is that almost a third of the children had significant change in their management, despite being in a region with limited resources. So not only is it possible to do whole genomes, but it's also possible for these regions to have the benefit of the whole genome. And this is an impressive slide because there are so many programs now trying to look at whole genomes in their populations, but this also reflects the bias. You see the areas that have population programs are those areas in the higher economic brackets We need to change this. We need to have population genomics in areas like South America, like Africa, Indonesia, that so we can get a better full representation. So this shows a great advance and a lot of activity, but it also shows that we're without an intervention, we could still have the same biases in the information generated. I'm going to step this side for sake of time and end on this slide where, as we think about, how do we learn about the human genome? There's a thought slide where, as you sequence more genomes, you understand and can identify more of the alleles and the variants driving rare disease. And we're on a path to really understand rare disease in a way heretofore not possible. But as we think about the human genome, for many of us, there's incredible opportunity with common disease. Yesterday, there was discussion about polygenic risk scores, pharmacogenomics, et cetera. We need many more genomes, and we may need to capture the full diversity to know how to bring that insight to human health. So I really applaud G2MC for its effort. I really appreciate the opportunity to present today. And with that, I'll end.
Well, thank you. Thanks a lot, Phil. And well, great presentation. I think we can now go directly to the panel discussion. I think I would like to start the discussion with some of the themes that you all, in a way or in another one, repeated. And is if we were really to go into advancing and thinking about your last map that you presented with the world map, where we clearly see this emptiness in cohorts in the south, and same what Ricardo say and Jeff, and in terms of education, we need to build this capacity, not just to rely on the capacity that has been built. And as Ricardo put it very nicely, it's just basically going from being customers are basically being producers. And how do you think, and well, I have to say that this is what we exactly are seeing with the COVID. There's no capacity to generate any of those tests or the reagents or anything here in the South. We have to rely on, most of us, importing, bringing those tests, bringing these technologies from the North. So I would like to hear from the panel on how you think we can build that capacity here in the South.
Okay, I'll jump in, and I think what we're doing today and yesterday illustrates an enormous opportunity that that maybe we have just passed over in thinking about how to engage young investigators and early stage trainees in developing as a workforce for genetics and genomics. What I'm talking about is really this virtual platform that we're using, at least as a way to achieve a greater degree of outreach than we ever have been able to achieve before. So I would at least urge us to consider that this crisis that we're in has actually catapulted us into using innovative and novel ways to reach communities that in the past we might not have been able to. So I'm going to just suggest that this is one of those modalities that could actually distribute the knowledge in ways that we haven't done before.
Yeah, and I guess my comment would be is, from a technology provider, I guess I always am concerned about placing, technology in an environment where there's not the ecosystem to support it. So I think, the comments, the presentation by Dr. Haspel about educating, about, building that community and that experience is so important. As far as making sure that we do bring this in country. Again, I represent Illumina. For those of you who know me, I spent 15 years in academia, past 7 years in industry. I can tell you from Illumina's perspective, we are highly motivated to ensure that these countries can build that capacity over time, can keep the data in country, can leverage it for the health of the country, and can, start being producers of the data, can start, being being a positive, very positive force, not only for the health of their communities, but also for the economy of their countries.
Could I just quickly follow up on what some of the things that Phil said, just to applaud H3 Africa as a paradigm of that, from the Wellcome Trust, the NIH, and other funders to bring both education as well as technology capacity to regions of the world that previously didn't have them. I think there are some H3 Africa representatives at this conference, but my observation is that's been a real catalyst for that part of the world.
Yeah, one of the questions that's also coming here through the chat is what we often see as, again, one of the challenges. One is to build that capacity in terms of technologies and in terms of our capacity to produce the reagents and all that. But the other one is and that was actually addressed very much by the World Economic Forum in the policy document they're developing to address the managing genomic data in developing countries and emerging economies. And is the challenges we have when North-South collaborations where samples are leaving the countries in the South and are being used and published. So, of course, there have been many, I would say, horror stories on that. And on your perspective, how you think we can really go beyond that North-South collaboration and really where samples are leaving and to really get, a more even level type of collaboration? Maybe Ricardo, if you,
sure.
I know you're very keen on collaborations in the South. But I think at one point we still need to rely on those North-South collaborations for some of those things. And how can we make those North-South collaborations much more productive for the South?
Sure. So I think right now, if you are in the research arena, the place where the actual analysis of the sample is occurring, maybe I think you need the local capacities, but maybe you need to be cost effective. So sending your samples to the US, Europe, or China, maybe it's not so terrible if you're able to be part of the second part of the project that is being part of the analysis. That's really important. If you can develop bioinformatics capability and having people capable to do primary and secondary analysis in Latin America and getting the right training to do that in the research setting and of course, I hope in the future in the clinical setting, that will be a game changer, I believe. So having access not only to our data, but also right now you can have access to data from many parts of the world. For example, I work in oncology and having the Cancer Genome Atlas made a whole difference, not only for the US and Europe, but also for everybody to jump into the analysis of complex genomic data. So of course, doing more analysis locally is nice, but being part of the bioinformatic analysis, secondary, primary, is very important for the development of capabilities in Latin America.
Yeah, and the model that we see this working towards is that of federated databases. So where in country, the investigators who are sequencing, they have control over their data. They can make sure they have that data, whether it's stored on premises or in the cloud. They have the right security and management of that. But if they choose to participate in international consortium, there's a model where they can provide access and maintain control of the data without sending the data or sending samples outside the country. And there's very good data that advanced analytics are just as effective in a set of federated databases rather than one big aggregate database. I think that's really exciting work that we'll see empower investigators in regions in the South to be able to build and participate meaningfully in collaborations.
Thank you, Phil. There are a couple of questions here for Rich, in particular two areas. One is it possible to maybe translate or use some of the materials that the ISCC is developing that could be used in Spanish or in Portuguese, that could be used in developing countries or in other areas, Latin America, Africa. So one thing is, can those be used? And as far as I understand, and some people ask questions here too, I'm not sure if there's any developing country that is part of the ISCC and that is really part of that consortium in terms of education and developing education tools. So that's the second part. And maybe the third part of the question will be if tools that are developed in the South could be somehow certified or could get some review and certification by the ISCC. So 3 components to the question, Rich, if you're still there. One is if some of the tools can be translated and used in the South, if organizations from the South can be part of the ISCC, and if you could serve as an organization that could somehow certify the quality of some of those tools.
Oh, thanks. They're all really good questions. The first one is we would love to have someone who, well, more for the second one and the first one, we would love to have members from any country are welcome to ISCC. And if someone, for example, from South America would want to translate some of our materials, that would be terrific. So we just need someone to do it. To be honest, I am working on my Spanish. I'm taking an online course, but I'm not quite up to it at this point. So, so it would be great if or any country, not just Spanish, whatever, we would totally welcome someone, and I would help with that as well as best as I could. In regard to credentialing, that's kind of a complicated question. It's not something ISCC or NHGRI could do. That's usually done by, government bodies or state bodies to credential. There are things like certificates that are offered, and there has been some discussion regarding that. It's also to get really credentialed. I see the question, something like genetic counseling. I mean, those are, that's not just a couple-hour online course. So we've mainly been focusing on ISCC as smaller, not major courses like that. So someone could search online for what's available, but things that often have credentialing of some sort cost a lot of money. But it's something I can bring back to the ISCC group, and I think there has been a little bit of talk, and I would have to speak to NHGRI, is there some certificate mechanism or something like that? But it probably wouldn't be to the extent of, weeks and weeks of coursework. So I hope that kind of answers it. And I'll just reiterate, please, if you just search ISCC and NHGRI, the website will come up and how to become a member and everything like that. And if anyone has any questions, they should definitely reach out to me. Thanks.
Thank you. Thank you, Rich. A couple of questions are coming here for Phil in terms of the cost of the regions in the South, because of course there's not even here possible to buy regions directly from Illumina or other producers. So is Illumina, or are you thinking about any specific programs or initiatives where there will be maybe a different pricing or a different way of giving access to these technologies, regions, to countries in the South?
Yeah, it's a very good question, and it is a challenge for us because, while we're a global company, we're a company of 9,000, and the globe's a big place. And so we do work through distributors in some areas like South America, like Africa, and some of the work with those distributors can be challenging. And so I think that has been called into attention to during COVID, where some of our efforts have been slower than we would like in responding to the challenge of COVID. I can't say there's a specific program, but we remain, foundationally focused on making sequencing affordable. We understand there's still limitations, and we want the affordability to extend across the globe. So it's a very appropriate question. We have been challenged to make sure that, it's a little crazy. I was recently had the opportunity to visit Ethiopia, Morocco, Egypt. In Morocco, right across the Strait of Gibraltar from Europe, they are charged 4 times what their colleagues in Spain and France are charged, and that's a big problem. And so we recognize it, and we're working towards addressing it. But I'm unfortunate at this point, I don't have a specific program. We do offer, grants. We do engage with groups, and specifically when we're working toward understanding how to bring populations together through collaborations like G2MC. There are opportunities that we can engage meaningfully and more directly to make it more affordable.
Go ahead and unmute yourself, Ligia.
Thank you. Well, first of all, it will be great to be able to use and translate some of the resources from ISCC and others to Spanish and Portuguese. We have a lack of material in education in genomics and medicine. So thank you. And just to follow up on the issue of collaboration and the inequality, this is something we are really worried in our genomics efforts in Brazil, so that we are not, once again, just generating the data and the intelligence and the discoveries are made elsewhere. And so what we're proposing, the approach is to give privileged access to the data first to Brazilian groups or groups that are collaborating with Brazilian groups, so that we can also learn and get back expertise to transform data in discovery and eventually in products. So that was, that's exactly why we're trying to implement that.
Can I make a comment on that? I appreciate that, and I've seen a lot of talk in the chat about this issue around equality in research, and I think this is one of the principles that we embrace for the G2MC, and particularly as we're trying to, I don't know what the right word is, level the playing field or make it equal footing for all collaborators, both from LMICs and non-LMICs. So I think one of the principles you espoused makes a lot of sense, but I think there should be also more opportunities not to upfront engage in the same rules of collaboration that we do with other colleagues, with co-authorship, co-grantsmanship, whatever it takes to make everybody feel like they're part of a team rather than sort of a transaction, which I think is what I'm hearing people think that this is. So I really appreciate the comments, and I think this is an area that we as a convening organization and a neutral party can really help perhaps make the rules of the road a lot more clear, so that LMICs feel like they're really getting what they deserve in their contributions to the science.
Yeah, and maybe adding to that, Jeff, many of the questions are also touching on points that we have been discussing at the G2MC inside the policy group, and in particular with the World Economic Forum, where we are, the World Economic Forum has developed a couple of vignettes and concrete examples of North-South collaborations and how to frame those in a way that are fully respecting and fully empowering the South to get the maximum of their research, instead of just what's happening in some cases in the past. So I think that there is great potential, and as you say, G2MC really has this mandate at its core, and is something we want to promote. Well, last meeting was in Cape Town. We could really discuss what's happening in Africa, and we see what's happening here in South America. There are many challenges that are shared and similar situations. So I think with that, if there's no more comments, Jeff, from your side, I think we can maybe go into a half an hour break.